Calcium signaling. Calcium signaling. Calcium signaling.
Did you know that cell signaling is one of the most fundamental processes for biological life? It allows for the flow of information from the cell surface to the cytosol and finally to the nucleus. It usually occurs as a response from the extracellular environment, which allows for the development, growth, and immunity and many other fundamental facets for biological life to occur. There are many types of cell signaling, but calcium signaling is one of the most important kinds. If we're going to talk about calcium, we should first talk about where it comes from. Like all elements that aren't hydrogen or helium, calcium is created by a nuclear fission in the stars. When these stars are big enough, they form supernovas, sending calcium flying into outer space. In the Earth's crust, calcium is the fifth most abundant metal, and plants take in this calcium through their roots. So we get our calcium in our diets by eating leafy green vegetables, nuts, seeds, fish, and of course, dairy products. Each person has around 1 kilogram of calcium in their bodies, which we use for a lot of different processes like cell signaling, cell division, fertilization, nerve signaling, and many, many, many more. Calcium ions act as a second messenger, so they can diffuse to other sites within the cell. Unlike primary messengers, which can only bind to unique receptors, secondary messengers can stimulate a variety of activities. The calcium ions are controlled by pumps and channels. This helps to make sure that calcium concentration inside the cytosol is much, much, much lower than outside the cell or in the endoplasmic reticulum lumen. The calcium concentration inside the cytosol is around 10 to the negative 7 molar, and its concentration outside is approximately 10,000 times bigger. Having the low cytoplasmic concentration is necessary for the cell because when the concentrations are deliberately increased, it activates important processes of the cell, such as apoptosis, transcription, and many more. To understand more about the mechanisms of calcium and normal cell division, let's hand it over to my friends at Calcium HQ, Jinsu and Joseph.
Thanks, Nicole. Here at Calcium HQ, we pride ourselves on all things Ca2+ related. Whether a cell divides, differentiates, or even dies is all dependent on cytoplasmic signaling and whether they activate or repress certain genes in the nucleus. One family that loves to help calcium-dependent signaling are the transcription factors myocyte enhancer factor 2, or MEF2 for short. MEF2 hang out loads in neurons and muscle cells. They're regulated by their pals MAPK, calcium-responsive enzymes that have free signaling cascades which start cell growth and differentiation. These star players include extracellular signal-regulated protein kinase, ERK5, C-Jun N-terminal kinase, JNK, and P38 kinase. One MAPK friend we're interested in is P38 kinase. They give MEF2 a big ol' hug in a process known as direct association. Once associated, P38 kinase will phosphorylate the transactivation domain of MEF2 to increase transcriptional activity. This interaction is spurred on by calcium so that P38 can get MEF2 to do its bidding. Want to know more about P38 pathways? Let's pass the baton to my colleague Jinsu.
Thank you, Joseph. So like Joseph said, MEF2 loves to help calcium-dependent signaling pathways, specifically P38 kinase. So let's dig more into this P38 pathway and its association with cell cycle and cell differentiation. P38 has 4 identical twin subunits who are P38 alpha, beta, gamma, and delta. This P38 family is very close with cell cycle and cell differentiation. In their relationship with cell cycle, especially P38 alpha is involved in the cell growth, and overexpression of P38 alpha leads to significant slowing of the cell proliferation. Furthermore, the family P38 loves to participate in the G1 phase of the cell cycle. The G1 arrest of NIH3 T cell was found to be P38 alpha dependent. Also, the regulation of P38 substrate, HBP1 and P21 also propose a strong link between P38 and G1 cell cycle control. Not only that, the P38 family loves to be involved in the G2 to M phases of the cell cycles too. The extrovert P38 alpha is activated upon M phase arrest by destruction of the spindle with nocodazole. In addition to their relationship with cell cycle, this P38 family is very involved with cell differentiation as well. P38 alpha and P38 beta are the important players in the cell differentiation for certain cell types, as they are required for the differentiation of specific cells into adipocytes, neurons, as well as myoblasts. However, sometimes things go wrong. Down in the cancer department, our colleagues Ashley and Arshvir have been hard at work on uncovering some of the missteps this process has.
Thanks, Jinsu. Similar to many processes, when something goes wrong with calcium signaling, there are often biological consequences. In healthy cells, low concentrations of calcium are seen inside of the cytoplasm, and high concentrations are seen outside of the cell. Maintaining this gradient is essential for proper calcium signaling. However, this gradient can be made abnormal through changes in calcium channels, which push calcium into the cell, or in calcium pumps, which push calcium out of the cell. One specific consequence of altering the gradient of calcium between the inside and outside of the cell is the development of cancer. This can be caused through an overexpression of calcium channels or through an underexpression of calcium pumps, which will increase intracellular calcium levels, increasing calcium signaling. Cancer cells rely on this increase in signaling to carry out several functions: migration, invasion, immune cell evasion, and proliferation. Specific calcium channels and pumps may be related to specific functions that cancer cells carry out. Evidence has indicated that invasion and metastasis may be promoted through an increase in calcium signals from specific channels such as TPC and IP3R3 in breast cancer. There are too many calcium channels and pumps that I could talk about that promote breast cancer. Let's pass it on to Arshvir, who's going to discuss a specific mechanism in cancer promotion through calcium signaling.
I love milk. Calcium, yum. All right, thank you, Ashley. One specific channel that has recently been shown to be upregulated in breast cancer is the mitochondrial calcium uniporter, or MCU for short. The MCU is responsible for mitochondrial calcium uptake, which triggers things like ATP synthesis, needed for many cellular energetics. Too little ATP in cells can induce autophagy, and too much ATP can cause organelle dysfunction. This is one of the many reasons mitochondrial metabolism is important and why deregulation of cellular energetics is considered an emerging hallmark of cancer. Studies have shown that the downregulation of MCU by silencing the gene in breast cancer results in significantly reduced cell motility, invasiveness, tumor growth, lymph node infiltration, and lung metastasis. The tumor microenvironment in cancers is very complex and often hypoxic. This stabilizes the hypoxia-inducible factor, HIF-1 which leads to the transcription of genes required for tumor progression. Mitochondrial reactive oxygen species, or MROS, act as important intracellular signaling molecules. MROS are crucial effectors of cancer progression and have recently been shown to upregulate HIF-1. Furthermore, HIF-1 in cancer cells induces the expression of glycolytic protein isoforms, and other than being well known for reprogramming cell metabolism, these glycolytic isoforms also participate in cell survival processes like inhibiting apoptosis and promote cell migration. In MCU silenced breast cancer cells, the production of MROS and the expression of HIF-1 were significantly reduced. This suggests that MROS and HIF-1 are involved in signaling cascades downstream of mitochondrial calcium uptake. Lastly, the rescue of HIF-1 expression in MCU silenced breast cancer cells restored migration and tumor progression, highlighting the importance of this hypoxia-inducible factor and the mitochondrial calcium uniporter's job in regulating its expression. Finally, we'll bring it to Ethan to unravel some cool future directions science is taking towards calcium mechanisms.
Woo! Wow, we sure know a lot about calcium signaling, don't we? But we don't know everything, and as scientists, it's our job to find out what we don't know, and then to study it. For example, studying intracellular calcium changes would tell us if what we've observed actually causes cancer, or if it's just associated with it, and that's really important. Additionally, in cancer, it's not just the cancer cells that go rogue. The cells around them do too, and cells like cancer-associated fibroblasts help cancer grow and spread. Calcium signaling might be involved in this process, but we simply don't know because we haven't studied it. And we think it would be really interesting to study it, and who knows what we could find and how it could help people. Maybe we'll find a new way to diagnose and treat cancer, and well, wouldn't that be great?
Thanks for watching.